Now that we've discussed the specification of the dorsal ventral axis, let's look at how different regions across the anterior-posterior axis are specified. In other words, let's look at the factors that allow the formation of different structures from the head to the tail of the organism. And we'll focus on the patterning of the nervous system across the anterior-posterior axis. The specification of this axis depends on a gradient of Wnt proteins, with the head having the lowest concentration of Wnt. In vertebrates, the forebrain, the hindbrain, and the spine regions of the neural tube have to be properly organized in an anterior-posterior direction. So the organizer tissues not only induce the neural tube, but also specify those different regions of the neural tube. And that happens in a region-specific way. And what this means is that more anterior portions of tissues derived from the organizer will induce the formation of anterior neural structures, while more posterior organizer-derived tissues induce the formation of more posterior neural structures. In this experiment, different regions of the organizer were transplanted from a late gastrula to the same location of an early gastrula. It was shown that the most anterior portion of the archenteron roof, which is the primitive gut, that contain head mesoderm, which is the anterior portion of the organizer, induce the formation of balancers and portions of the oral apparatus, and these are anterior structures. The next most anterior section induced the formation of different head structures, including the nose, the eyes, and otic vesicle. The third section, including the nodal core, induced the hindbrain structures, and the most posterior section induced the formation of the dorsal trunk and the tail. Recall that the identity of the dorsal blastopore lip changes over time as the more anterior structures begin to involute during gastrulation. So if you were to transplant a dorsal blastopore lid from an early gastrula from one salamander to another early gastrula, you would end up with a salamander that has a secondary head. Whereas if you were to transplant the blastopore lip from a later or more advanced salamander gastrula to an early salamander gastrula, you would end up with a salamander that has a secondary tail. This is because in the early gastrula, the blastopore lip is composed of the anterior endodermal and mesodermal structures, which induce the anterior ectodermal structures, the anterior neural tissues. But later, the blastopore lip is composed of more posterior mesodermal structures, which will induce the formation of more posterior neural structures. These results show that the first cells of the organizer to enter the embryo induce the formation of brains and heads, while those cells that form the dorsal lip of later stage embryos induce the cells above them to become spinal cords and tails. Given these results, the question then became, what are the molecules being secreted by the organizer in a regional fashion such that the first cells involuting through the blastopore lip, the endomesoderm, induce the formation of head structures, whereas the next portion of involuting mesoderm, the nodal core, induces the formation of trunk and tail structures. Well, the first tissue to involute during gastrulation is the tissue that becomes the pharyngeal endoderm and the precordial plate mesoderm, and together these are known as the head endomesoderm. In addition to secreting BMP inhibitors to allow the formation of the nervous system, these tissues secrete inhibitors of the Wnt pathway. And blocking of the Wnt pathway is necessary to allow the head to form. So in regions where the head structures form, both BMPs and Wnt's have to be blocked. In the region that will become the trunk or the spine, the nodal core secretes BMP inhibitors to prevent the ectoderm from becoming skin, but it does not secrete Wnt inhibitors. Since inhibiting the Wnt pathway is important to allow the head to form, if you were to overexpress Wnt inhibitors, the embryo would end up forming extra heads or ectopic heads. In this image, the embryo was injected with an mRNA for the Wnt inhibitor Cerberus. As you can see, the embryo developed an extra head as a result. If instead you were to block Wnt inhibitors like Cerberus, then the head would not be able to form. Besides Cerberus, there are other Wnt inhibitors that are synthesized predominantly in the endomesoderm cells beneath the prospective brain. If embryos overexpress these inhibitors, Wnt signaling is blocked throughout the embryo and the embryo becomes all head, lacking all posterior and ventral structures. So if blocking wind signaling is needed to allow the head to form, it only makes sense that the presence of wind would be responsible for allowing the tail to form. And that's precisely the case. Winds act as one of several posteriorizing molecules. In Xenopus, there's a gradient of wind signaling and beta-catenin, with the concentrations being highest in the posterior and absent in the anterior. And if that gradient is altered, the location of those different neural structures changes. So there are two major gradients in the amphibian gastrula, a BMP gradient that specifies the dorsal ventral axis and a wind gradient specifying the anterior posterior axis. This figure summarizes the factors involved in the establishment of the dorsal ventral and anterior posterior axis. These factors, which include wind and BMP inhibitors, lead to the formation of wind and BMP gradients. However, it's important to remember that both gradients are established by the initial gradient of nodal paracrine factors and the presence of the transcription factor beta-catenin in the dorsal vegetal cells. In this figure, it's also important to point out that while winds are important for specifying the anterior-posterior axis, there are other factors involved as well. Fibroblast growth factors appear to be critical in allowing the cells to respond to the wind signal. Retinoic acid, or RA, also forms a gradient that is highest at the posterior end of the neural plate and is important for the patterning of the hindbrain. As we'll see in more detail when we discuss mammalian development, retinoic acid acts by activating the expression of posterior Hox genes, and these genes are involved in the patterning of the anterior-posterior axis. In amphibians, RA is important for the formation of the tail, the most posterior part of the tadpole. Together, the posterior-anterior wind, FGF, and retinoic acid gradients function to determine the boundaries of the Hox genes along the anterior-posterior axis. We've looked at the specification of the dorsal ventral and anterior-posterior axis. Let's very briefly look at the specification of the left-right axis. Like humans, amphibians are not symmetrical. There are several organs, like the heart and the gut, that are not evenly balanced on the right and left sides of the body. In all vertebrates that have been studied so far, the key event in left-right axis formation is the expression of the nodal gene on one side of the embryo, on the left side of the embryo in the lateral plate mesoderm. And we'll talk more about this when we discuss mammalian development, but the key event that restricts the expression of nodal to the left-hand side is the movement of cilia. And these cilia are made of microtubules, which are cytoskeletal proteins, and other microtubule-associated proteins. As in humans and amphibians, mutations in tubulin, which is the protein that makes up these microtubules, or in genes that code for ciliary proteins like dynein, affect the movement of cilia and cause a reversal in the location of the internal organs.